My name is Charles Efriye Deborah from CSR Crop Research Institute. I'm a senior research scientist, agronomist, and a biosafety officer. So I'll be presenting on effective biosafety systems for nitrogen use efficiency, water use efficiency, and salinity tolerant. I think from morning we've been listening to mosquitoes, animals. Let's digress a bit, because when we talk about biocontrol, it talks about crops, insects, and the environment as a in general. So I'll keep it short. I didn't want to add jollof rice to the list or the pictures here for my brothers from Nigeria to claim patency. Oh. So we say that annually per capita consumption of rice is about 45 kg. That means by the end of the year, and I know a lot of people here through breakfast and lunch has eaten rice. In Ghana, we are importing about 50% of the demand and spending about $200 million every year on rice importation. So you can see from figure 1, figure 2, how acreages have been increased over the years in production and how yields too has been faring over the years. You can see that from 2029 to 2020, there was a dip. A lot of these wetlands are being transformed into other activities. You see wetlands, the little time you go there and either a church has bought the land or they're using it for other activities. So we continue to reduce in acreages, therefore affecting the yield that we. So this is our contribution to rice production. And you can see rain-fed, lowland, upland, and irrigation. And you can see the average yield from 3, 1.5, and 5 tons per hectare. So rice production or cultivation has had some challenges, like the socioeconomic factors, that's lack of credit, high cost of inputs, high cost of labor, and lack of mechanization of activities, and biotic stress like blast, RYMV, and abiotic like poor soil, fertility, drought, and salinity. And you can see how this rice is doing. Next to it is a rice that has been affected by RYMV virus. That means you look at these ones too, you can see how bad that some of the fields are looking as a result of nitrogen deficiency, drought, salinity effect. Therefore, we have to choose the correct variety which can withstand some of these stresses that we're talking about. So you can see here, these 2 different rice varieties were planted at the same time. You can see how one is doing very well after experiencing a little drought. So effect of these abiotic stress, like the nitrogen deficiency, can reduce yield up to 90%, drought, total devastation, and yield loss of up to 50% due to salinity has been reported. So when you talk about the newest rice, it was a project which were in 2 types of products, the NUE and the one having all the 3 genes in there. And they were Nericafol, which is known to be an upland rice, variety from African rice. And the genes that were transformed were as follows. So it was a USA project which was coming from Seattle, Colombia, and the genes were transformed at Acadia in the United States. And this experiment took place in Ghana, Nigeria, and Kenya, and Uganda. As time goes on, Kenya, Uganda, and Nigeria stopped because they were not doing very well. It was only Ghana that was providing data to AATF. So how did we start our biosafety system? So it started by meeting our sector minister, and that is when those time it was the National Biosafety Committee that transmogrifying into National Biosafety Authority. And that was when we started our, our regulatory framework. That is the National Biosafety Framework. Then we started the Act and the rules of Biosafety Authority. And all this was done in guidance to the international protocols. And we also adhered to the institutional compliance. That's the CSR. We have an act that governs science and technology. So the experiment was done. 5 trials were done involving the 16 lines. And we did 2 regulatory trials done on the lead NUE lines, where we did a food and feed safety analysis, environmental risk assessment. And we, under the risk assessment, we evaluated potential effect on non-target organisms, soil and water quality monitoring, and we did human health assessment and socioeconomic consideration. So let's look at some of the biosafety, and these are not very common when you visit a lot of CFTs. So we had our physical containment, which protected our confined field under lock and key. And I stood in front of the security gate, so you will see the security gate. Then we also had our biological containment. That is the use of sterile GM varieties where applicable, where we also use the isolation distance and temporal separation. We also did control field trials, that is limited release protocol, and we had inspectors, that is the NBA, visiting us and external reviewers visiting our facilities to know that the right thing is being done. And I remember when we were constructing, they used ninja resin wire, and as a biosafety officer, I had to visit. And I told them, if you build it like this, you are inviting a lot of mice. So we have to use a small, I think it's 1 cm by 1 cm squares, and we have to bury it about a quarter of a feet down around to prevent mouse from getting into our field. So we did our regular monitoring, that is pre-trial monitoring before the trial. We did during and post-monitoring. So a lot of education has gone in getting communities involved in everything. So you look at the plate 6, you could see that the community together with the PI, the scientists, the chiefs, everybody came. In fact, when this thing was brought first and we opened it, the chiefs were like, it's the rice, the normal rice that we know. They thought the GM rice was something different from what they know. So they didn't even spend time with us. Oh, it's rice. So you know how to go about it. And as a biosafety officer, you have to make sure you stay throughout the planting season, and you could see how they were performing at vegetative state, flowering, and you could see our facility was used as a study area where we could invite journalists, and just as we had communication people joining us for this, always they can tell the story if you educate. You know, scientists, we are not good at telling our stories, because you sit and you'll be talking about DNA, and what is DNA, but they know how best that they can. So we always invite them to come look at what is going on and tell the story which the public can get. So we had farmers, policymakers, scientists, and consumers coming around, and that was to also look at the public perception and acceptance of this GMO. And we've had a lot of trainings, that is staff training, public awareness, that is community sensitization on the GM technology, and media engagement. So we had our PEI going through all the process in data collection, vegetative stage, and samples collected for the food and feed analysis. And some of the things that we normally go, as I said, biosafety, effective biosafety, and you see the A having our former CEO, Eric, inspecting the package that was received to confirm that it has not been tempered before giving it to the PI. And there was one instance I received it and gave it to the PI. Together with the PI, we went through to see that whatever that was brought or written on the paper is exactly what was in the container. And we had the movement book, what goes in and comes out every time. And you can see some of the things that we do to make sure that there's effective biosafety system. We normally burn the remaining ones after every cycle, because when it moves to one stage, normally we receive fresh seeds. So we make sure that we don't put them there for anybody to pick them out before the final assessment can be done. So a bit of results. So the lead NUE has a yield advantage of just 15 to 30% from our trial. And we say the lead NUE was also similar to that means that comparing the conventional and the GM, there were a lot of similarities. And the other analysis done didn't show much difference. So what is the next step? We prepared the dossier to the National Biosafety Authority, and at the end of the day, the sponsor said that they are looking at between 30 to 50% yield. We were having 10 to 15, and therefore the experiment cannot go on. But these are just some statistics to look at the benefit of the technology and expected benefits that we were looking at, that the poor farmer could afford the recommended level of fertilizer, and also to improve livelihood. The effect on climate change too can be mitigated, and marginal areas which cannot support rice could also be cultivated using this line. I want to say thank you to all donors and the National Biosafety Committee. Last year, we were fortunate to be the best IBC, and we're given an award by the consortium. Thank you. And that's the picture for you.